who's also on the design team here, is Darin Taverna, and we're delighted to have him here. Darin is the current Director of Computational Biology at Systems Imagination, Inc., a new biomedical analytics startup redefining how pharma and physicians get insights from data. One of the themes of our project is bioinformatics, which might be a new term for you, but it's use of giant databases, as you can imagine how much information we're now trying to synthesize about cells and molecules and patients, as well as the use of computers and programming in the biolabs. So Darin is also the adjunct professor at Phoenix College in biology. Before he moved to Systems Imagination, Darin spent 10 years at TGen, the Translational Genomics Research Institute, studying the genetic basis of human disease. He has a Ph.D. and a master's in biophysics from the University of Michigan, and he got his bachelor's degree in physics from Carnegie Mellon University. And I always have to say this, but very exciting development is he's one of the 3 finalists in the Arizona Republic's top home chef competition. So he's a very talented young man. So Darin.
Hi, so my name is Darin, and I'm going to talk to you today about personalizing biomedicine. I am a computational biologist. A computational biologist takes algorithms, models, knowledge of physics and math, and applies them to solving problems in biology. And what really got me interested in this is as a physics undergrad, I took some biology courses for fun. And when I was reading through our textbook on developmental biology and saw this page, I really got excited. And because there weren't any classes for computational biology back then, not that anyone told me about anyways. And so this was on cell sorting. And so the way to look at this is that you have A and B cells. And the A cells have a certain interaction, and the B cells have a certain interaction. And there's also a different interaction between the A and B cells. And so it turns out that you can describe how these cells will arrange themselves by the interactions between the cells. And so on the right-hand side, you see, it's very simplified, but they're thermodynamic equations for how these cells interact. And so just depending upon that, you can get cells that sort together and form a structure. You can get random mixtures of cells, or they can just separate completely. And what was exciting for me about this is the implications that it had of combining biology and maths, and how fundamental it is. This is the basics of how life begins, how life can separate out into different structures that then become different systems and organs. It's also how life can end. If you have cells that don't communicate well with each other, you can have cancer, you can have cells that metastasize and move around the body. So it really, the implications of that as an undergrad, it was just fascinating for me, and I never looked back. I mean, I just kept exploring this area. But as far as the talk today, I wanted to talk about where we are now with personalizing biomedicine. And so there's really 3 areas that I'm going to talk about today. There's certainly more, but I'll talk about 3. Personalized genomics, which is knowing your genome, getting sequenced and knowing all the variations within your genome. Personalized diagnostics and risk assessments. So that would be taking tests that use information in your genome, among other things, and predict what risk you have of getting a disease, or maybe identifying if you actually have a disease. And then finally, personalized medical treatments. So using information from the previous 2 and just general knowledge about medicine to get the best treatment for you. If you go into the doctor and you want a personalized medical treatment. So I'm going to argue right now that personalized genomics is a solved problem. As of the beginning of this year, companies that do sequencing for folks came out with the pricing of $1,000 genome. You can get your whole genome sequenced for $1,000. And this is amazing because when it was done the first time they sequenced the human genome, it cost $3 billion and took 10 years. Now it costs $1,000, and you can do it in a day or two. It's just fantastic. So I would close the book on that. That's done. But what about personalized diagnostics and risk assessments? Where are we with that? Well, I thought, what better than to talk about my own genome? So I actually have a list of variants here. SNPs stands for single nucleotide polymorphisms, but they're all variants. So this list is just a clip of my genome and just the variants in the genome, but this list goes on for millions of variants. I thought it was interesting to show to you today because this particular SNP in blue here says that it's linked to studies that show that I might have a 24% better long-term memory than the average person that doesn't have a C and a T in their genome. However, I also have another variant, which isn't on this page here, that says I have a poorer than average short-term memory. So, what do you do with that? I think about it, I process it like, it may take me a while to get something in my head, but once it's there, it doesn't leave. Or as my friends would say, I'm stubborn. I can also take this information and compare it to information on drugs and how drugs interact with genes. So it's summarized here, but all these SNPs on the side here have information about how that variant would change a protein that these drugs would interact with. And so again, it's a summary, but you can get some idea looking at this more in depth, how a specific drug that you might get would interact with your body, how effective it would be, how you would respond to it. If you had two drugs that may seem the same, that cost the same, that have the same side effects, you could look at something like this and maybe say, what would be better for me? But it's not perfect, and nor should it be. As you've heard today, the genome interacts with an environment. There's this genomic part of life, and there's this environmental part of life. And also there's just plain randomness. So, what the details of these studies that have all been done give you a risk score. They give you a propensity that you might actually have a characteristic. So for me, it says I should have an increased risk of getting blue eyes, and I have brown eyes. It says I should have male pattern baldness, and not quite. Not gonna win. But there's other things. There's harmless things like hair and eyes and whatever. But when it throws something to you like this, I have a 1.3 times risk of getting prostate cancer. You wonder, what is this? Like, how much do I really believe this now? Because this is more serious stuff. I mean, I have a father. He's in his 60s who already went through prostate cancer surgery. So the average risk of the males in this room getting prostate cancer is around 15%. A 1.3x risk would mean I have a 20% chance. So it also has some information in the studies that were done that say aspirin can reduce the risk. So, it's just something to think about. And it's not just me. Almost a year ago today, Angelina Jolie came public with the fact that she looked at BRCA1, a breast cancer gene, and decided, based on her family history, the fact that her mother died very young of breast cancer, and whatever was known about breast cancer at the time, decided that she had an 80% chance of getting breast cancer in her future. So she had what I think is a very brave thing, and she had a double mastectomy done, preventative. And if you look at, let's see if I can get the laser, it's not really showing up. If you look at the center of that pathway there, this is a DNA repair pathway. BRCA1 sits right in the middle of that. It's a very key gene. And if that's got a variant in it that messes up its function, it's very central to the process, and it's going to mess up everything else below it. But that's, you have two copies of every gene. And this is this is what we know about the pathway right now, but we're still learning things about the genome, and there might be other parts of this pathway that are yet known that compensate for BRCA. She did the test on BRCA1, and there's plenty of other elements in this pathway, so it could be that she had yet another variation that maybe would make it a 90% chance that she had breast cancer. So it's just, you really have to be well informed when you start thinking about what the implications of these tests are. So where are we right now? I think we're there, but we should interpret with caution. So I know it says December 6th, 2014. It's a forward-looking talk, but not that forward. It should be 2013. So just at the end of last year, the FDA said that people that do this genomic reports should drop claims related to health and disease. Just for that reason, they felt that the tests weren't informative enough for the consumers. So now we're down to personalized medical treatments. Where are we with that? Well, that's where my work comes in. So there's a lot of information I left out. It's not just the genome. You have how things are transcribed, how proteins interact, how drugs interact, how things are metabolized. There's so much there. And so we're trying to actually connect that in a way that would make doctors have better decisions in the clinic, that pharma can design better drugs for you. And so I don't think we're there just yet. And I don't want the X to be negative. It really, it means we have work to do. And so there's limited success right now in clinical trials. Patients are living longer. TGen did a study recently where they found, on average, by doing personalized medical approaches to people that have failed traditional therapies, you can extend the life of some cancer patients by 3 to 6 months. But we're not there yet. It's still in the research phase. And I think we need to be able to merge data better, to search that data better, to combine it with prior knowledge better, and then also come up with better ways to validate those models. So we're not testing the patient. We want to test outside the patient, or whether it's a printed tissue or cell culture, and then apply it to the patient. So one way that we're working to deal with this is this graph over here represents a body of knowledge around medicine. Consider it everything that your doctor might know, everything a pharmacist might know. And what you want to do is you want to take your genomic information and take only the interesting connections in that graph. You want to filter everything else out and just look at it for what pertains to you. Now, in this graph is not only stuff about your diseases, but also stuff about like what hair color you might have and eye color, height, whatever. So you want to filter that out once again for a specific disease context. Okay, so there's a lot of filtering that goes on. And once you do that, then you can begin to personalize medicine. So how would I actually deal with the state of the art today? Well, I would monitor my diet, nothing too extreme. I'd monitor my diet. I'd be more prone to use aspirin as opposed to ibuprofen maybe, because it will lower my prostate cancer risk. Once I approach my 50s, I want to do more in-depth monitoring with whatever tests are available today. I have a better idea of what medicine will work for me. I'm not going to throw in my comb just yet. And if someday I have a kid with unexpected blue eyes, I'm not going to blame my genome. I'm probably going to question the mailman first. So, anyways, thank you.